Hi, everyone. Welcome to Cited, a Biotech and Health Extension Group sponsored by 100 Plus Capital. I'm so delighted to have Maria Cuervo here. I know that we've at least tried to reach out to you multiple times because you've been on our nomination list for quite some time, including for in-person events and so forth. So I'm so delighted that it finally worked out. And I know that many people here have been really looking forward to this. So thanks a lot for joining. I'll give it over to you just really in a second to hit the ground running. And yeah, if anyone has questions here in the group, I'll be in the chat. And now today is joining us to discuss targeting selective autophagy in aging and age-related diseases. And I'll share a little bit more about your bio here in the chat and your Zoom in for me at the time. So thank you so much for joining, and I'll be in the chat from now on.
Okay, so should I start with the presentation?
Yeah, that would be great.
Terrific. Well, thank you so much, Alison, and thanks everybody for connecting. I'm not very sure about the level of detail that you guys want, but Alison mentioned to get kind of a 20 to 30 minutes overview of what we do, and then we can discuss more. I put different things, and whatever you guys want to discuss, I'm always more than happy. So I think you might be able to see my slides right now. So we are very interested in targeting selective autophagy for aging and age-related diseases. And this group is already very familiar with different iterations of these hallmarks of aging or drivers of aging. And you probably have noticed that despite many versions out there, all of them coincide at least in one of the hallmarks, that is proteostasis. And that's the one, or loss of proteostasis as a driver of aging. And that's the one that we have been mostly interested. I'm not very sure how familiar you are with proteostasis, but just in case there is new people, proteostasis originates because when you look at the cells, the environment, the cellular environment, is a very, very packed environment. So this is, for example, a 3D reconstitution of inside a cell, and you have your organelles, your ribosomes, your proteins, your lipids. So everything is very close to each other. So in this environment, it's absolutely necessary that each and every one of your thousands of proteins that you have in the cell are at the right conformation, in the right place, and at the right time. Because otherwise they can really start interacting very aberrantly. And many of you might have heard about aggregates and how these toxic proteins start accumulating. So who takes care of this quality control of making sure that proteins maintain homeostasis? So the ones that take care of that is what is known as the proteostasis network. And basically they have two components. The chaperones that are the one checking to make sure that everything is in the right conformation, right place, and right time. And then if that doesn't happen, if a protein is wrong conformation or is misplaced, for the cell, it's more beneficial to eliminate it rather than trying to fix and repair and spend so much energy on that. So the ones that are taking care of this cleaning, of this elimination of these misbehaving proteins, are really the proteolytic systems. And there are two major ones in the cell: the ubiquitin proteasome system, that many of you might be very familiar, and the others are the autophagy system or the lysosomes. And by this cartoon, what I wanted to represent is that even I'm going to be talking about autophagy and lysosomes in the context of protein homeostasis or proteostasis. As you are all very familiar, all these drivers of aging are tightly connected. So autophagy is one of those processes that seems to be related with many of these drivers of aging. So failure of autophagy contributes to alterations in metabolism, alterations in stemness, changes in epigenetics very related to aging, difficulty to adapt to stress. So again, I'm focusing in the role of autophagy in proteostasis, but just think that when we target or when we modulate this pathway, we might have an impact in many other drivers of aging. So autophagy is the term that we use to refer to the degradation of any kind of cellular components inside lysosomes. And there are different flavors, different types of autophagy, depending on how you take something from the cell and deliver it to the lysosome. So you can put it in these vesicles, like the same way that you can take the garbage in different ways. So you can just wrap them in these vesicles that then fuse with the lysosomes, and this is what is known as macro autophagy that has different flavors. You can just create different invaginations, different vesicles or small vesicles from the surface of these organelles, the lysosomes, the endosomes, and these are called micro because there are smaller vesicles than these big ones that are macro. And then you can also be very selective and just choose proteins one by one. This is wrong, this is not wrong, and be very selective and deliver it directly to the lysosomes, cross the membrane of this organelle, and get degraded. And this process is what is known as chaperone-mediated autophagy. So in our lab, we really study all types of autophagy because they coexist. It's not that one of your cells is going to do one type of cleaning, the other is going to do the other. No, these systems are always present in all the cells, are always functional, and they crosstalk. They kind of decide who takes the garbage where. But I'm just going to focus for simplicity in this chaperone-mediated autophagy that I will call CMA for short, just because it's probably for the point of view of therapeutics and interventions and gerotherapeutics in particular, I think is the one that in our lab we have advanced a bit more. But then if you guys want to discuss about what are we thinking on how to intervene in the other forms of autophagy, I will be happy to go about that. So this chaperone-mediated autophagy, as I say, it's a very, very exquisite mechanism of degradation. Imagine this green protein is what you need to eliminate. The chaperone that will be here in red, recognize it, bring it to these lysosomes, and then you have kind of a receptor transporter. And this is the only one that I'm going to mention by name, that is this LAMP2A. So it's a membrane protein that is kind of the receptor for the pathway, but then it polymerize and forms a discontinuity in the membrane so the proteins can go through. So it's called a translocation complex, and then the proteins get degraded in the lumen. So CMA, or this chaperone-mediated autophagy, is present in all the cells constitutively, in all mammalian cells, but then it can also be induced in response to stress. So you upregulate it in response to multiple stressors, as many other components of the proteostasis network. And these are some examples of stressors that activate this pathway. The role, as I say, is mostly participate in quality control. Whenever you have a protein that is not properly full or that has been damaged, can be eliminated by this pathway. But also because these lysosomes, once they degrade these proteins, they recycle the amino acids. They break down the proteins in pieces, and whatever is good, they recycle to the cytosol. So this pathway also contributes to replenish the pool of your free amino acids in the cell. And then also something to keep in mind is that I'm always talking about garbage, right? But this pathway has the capability to degrade fully functional proteins that are not damaged at all, but it just eliminates them to terminate their function. So imagine an enzyme. You want to stop this process, so you can just eliminate this enzyme, so then whatever it was catalyzing will not occur. And the same, for example, imagine a transcription factor. You want to stop transcription, so you eliminate the transcription factor. And it's still fully functional, but you just eliminate it to terminate the function. So that makes this pathway to have important regulatory functions inside the cells. Then the other major reason why, we were interested in this pathway because when I was a graduate student, I contributed to the discovery, but I think what has kept us going through the years is the fact that very early on, we found that this type of autophagy decreased with age. So with the time, we have developed different mouse models. We do a lot of mouse work trying to be able to visualize this process because, all that I have told you, we learn it by biochemistry, by isolating these lysosomes and study this transport. But right now, we have developed these very nice mouse models where we have a fluorescent reporter that contains a targeting motif that when it's recognized by the chaperone, is going to bring this protein to the lysosomes following this pathway, this CMA. So then the protein goes there, and then the lysosome highlights, in this case in green, because it's a green protein. It's a dendra protein, but it happens to be green color. So then now we can look, for example, at the brain, different organs. So this is what you have coming here is the hippocampus of an animal. This is a healthy wild-type animal. And each of these little dots corresponds to lysosomes that are being highlighted because it's at there. They are going into a single cell. For example, this is a single neuron, and this is just highlighting the surface of the neuron. And then you can look inside, and learn where not only that CMA is occurring, as you see in these little dots, but where in the cell, exposing these animals to different contexts or crossing with mouse models of disease, we can learn when this pathway is altered. So something that we have started to do is, for example, now, thanks to the pandemic, we have a lot of animals that become old and we couldn't do experiments. So now we have aging animals, so we can study how CMA changes in every single cell type in every organ in the body. So I'm just showing you here the brain. This is work from Rabia. So if you look at a young animal, and I'm going to make it bigger, so this will be, for example, a neuron. I hope you see it here. And all these little dots, these are areas where CMA is being active. We haven't stressed this animal. This is normal activity of this pathway. And we decide to also do males and females. So this way we learn a little more about sex. That is something that I think we have unfortunately not paid so much attention until now. But, in this particular organ, this particular cell type, males and females have very similar CMA activity to start with. But then when you age those animals, so this is aging, and as you can see, these are neurons that you don't see these beautiful dots. You don't see your lysosomes being activated. So as you can see here, there is a decrease in activity with age, and this decrease is much more pronounced in the case of the males for this particular region of the brain. And by doing these experiments, we have also learned not only that there are differences in other organs, there are differences in the basal CMA activity depending on the gender, but also that the impact of aging is different depending on the gender. But the mechanism by which CMA decrease, you see here a decrease in both cases, the mechanism behind it seems also be different. And I think this is important, all these details, because if we want to fix a pathway, if we want to target it therapeutically, we have to know what is wrong. And it might be that it's not the same thing is wrong in males and females. So far, we have found something that is common for both of them and that is wrong. But, I think it's something to keep in mind that there might be opportunities of intervention that might be very sex dependent. And then, of course, until now, I have been talking to you about animals. So what about in humans? How are we going to measure CMA in humans? So something that we have been doing, and this is work from Matia and Adrian, they have developed an algorithm that allow us to infer CMA activity based on the expression of the network of the components of this pathway. Through the years, we have identified these 20 components, and we keep adding to this algorithm. And just as a proof of concept, we start data mining in RNA-seq data or proteomics data that has been published. And just to show you, for example, this is the brain, different regions of the brain. This is the nigra, and this is the prefrontal cortex from healthy all individuals from 40 years old to 80 years old. And as you can see here, this CMA score, this CMA activity, based on these studies, decrease in both areas of the brain. The patterns are a little different, but also we can follow almost in any organ where there have been some omics done. And we are in the kind of the omics, so we are taking a lot of advantage. So I'm just showing here, just so not everything is about the brain. This is, for example, the aorta. We have a study showing how CMA activity determines the probability of having a second incident when you have a heart condition, cardiovascular incident. So we were looking at the aorta, and it turns out that there is a big decrease in CMA activity with age, even in normal healthy individuals. If you impose bad diet or some genetic predisposition, this decrease is even higher. So I think this at least validate that this decrease on CMA that we always describe in animals with age, it also happens in humans. And then, as I say, there are probably different mechanisms behind this decrease of CMA, but something that seems to be, I will not dare to say universal, but it's one of the major changes that we see almost in all the tissues and all the cells, is a reduction in the levels of this receptor protein. The only one that I say by name, this LAMP2A protein, as you get old, these are, for example, still animals that 3 months, 12 months, 22, and you can see different areas of the brain. So there is a constant factor here that is always this decrease in LAMP2A, and that correlates very well with the changes in activity that we saw in our animal, the fluorescent, the one that has the fluorescent reporting. So based on this little introduction, basically our lab is interested in trying to figure out why these pathways, in this case CMA, do not work as we get old, or why they stop working. What are the consequences of these pathways not being functional? And then, of course, if we think that the consequences are important for aging. How are we going to fix it? So one thing that I'm going to talk about mechanisms is what I just told you, that CMA activity decreases with age, and in part is because as you get old, the levels of this receptor protein decrease. So basically, what we have tried to do is to understand what are the consequences rather than using an old animal, what many other things would have changed. We have tried to mimic this decrease in LAMP2A that's happening in lysosomes with age in a young animal. So basically, we have eliminated this receptor. And what we have learned, and this is a summary of many years of work now, but we have done tissue-specific reduction of CMA in a young animal. And what we have found, and just to summarize, is that you find phenotypes of degeneration. For example, in neurons, you have a neurodegenerative phenotype. We have work done in the retina. You have retinal degeneration when you don't have CMA or you reduce CMA, also muscle loss of function, as you can see by the central nucleoli. We also have metabolic phenotypes. So we have done work in liver and adipose tissue. And basically, you have many characteristics in these animals that mimic the metabolic syndrome of aging. They also have a loss of stemness. We have some work done with hematopoietic stem cells showing that if you don't have CMA, they age, they lose the ability to repopulate the bone marrow. The Mathian lab has described how when you don't have CMA in immune cells, you kind of develop immunosenescence. You have work showing that disruption of CMA results in alterations of circadian rhythms and sleep, something also very important for aging, and higher malignant and higher propensity to malignant transformation. So of course, as you see, these are many features that you see in aging, but these are young animals. These are 3 to 6 months old animals. So I don't want to say, of course, that CMA is the cause of aging. That is not a single cause, but at least the decrease that we see in CMA with age clearly contributes to the aggravation of some of these phenotypes that are characteristics of aging and some of this functional loss. So then I hope I have convinced you that this pathway is important enough, because when you take it out or when it decreases, you have all these manifestations that result in loss of function in multiple organs. So what are we going to do about it? All of us are going to get old and our LAMP2A is going to decrease. So can we fix it? The lab has really engaged in a three-pronged approach. We have done genetic approach as a proof of concept to convince ourselves that it's worth investing in developing chemicals to modulate this pathway. But also, I guess my MD part from time to time kick on. And if instead of having one more drug, I could have some dietary recommendations. And these pathways are very closely linked to diet. And it's something that I can also discuss. I mean, one of the mechanisms that activate these pathways, lack of nutrients. So I think there are many ways in which we can also interfere. But just to give you a glimpse of what we have been doing. So genetically, as I mentioned, we knew that one of the problems is that the levels of this receptor in lysosomes decrease as you get old. So what we decide, and this is work from Imma Tasset, what she decided is to create an animal in which whenever the endogenous copy of this receptor go down, she can activate an extra copy and maintain the levels as they were when they were young. So we are not going above what is normal CMA. We are just maintaining them per life. So the wild type CMA will decrease, but the animal that we have manipulating LAMP2A will have constant preserved CMA till late in life. So when you look at those animals, and those animals will be always in orange here. So this is just lifespan. This is in males and this is in females. And as you can see, we have an extension of lifespan of between 18 to 23%. That is pretty good for mice. But also more than the maximal lifespan, I'm very excited about the idea that, they have also a better mean lifespan, and they are kind of flattening a little the curve at this time. So we thought that this, okay, maybe this reflects better functionality, not only that they live longer, but that they are healthier. So in collaboration with Rafa De Cabo, we have done these whole panels of frailty. So basically what you have when you go to the geriatrician, but adapted for animals. But rather than show you 32 graphs, I thought it was better just to put the animals. So these animals have the same age, so it would be equivalent to 80 years old. This is one type, the guy here that is balding, has white hair, barely moves, is really anxious, is continuously grooming itself. If you look at the tail from time to time, very little mobility. And then this is his brother, but we just preserve CMA. And he's a happy fella. He's moving around, did cognitive studies, circadian rhythm. So, we are doing something good because the animal is way healthier. And it's not only that it lives longer, is much more healthier. And then I can tell you why we think this is it. But I think we cannot blame everything to have better CMA. This is our intervention. But as you know, all these drivers of aging are really highly related. So when you look at these animals, senescence, they have better preservation of the epigenetic traits, and we're doing that with the Steve Horvath. They respond better to stress. So basically, almost every driver of aging seems to be improved because of this interconnection. But then what else are we doing? So we are interested in trying to develop some chemical compounds that allow us to do that or to improve CMA in animals where we already have this decrease in CMA and hopefully. So basically, of the different approaches, I just wanted to comment in one because we have published it very recently. This is in a collaboration with Everis Karathiotis, an amazing chemist at Einstein. And what we found very early on is that CMA is under the negative regulation of this retinoic acid receptor alpha. So this is a physiological break on the amount of CMA that you can have. So we thought that, maybe in aging, if we take out this break, we might be able to upregulate CMA. So using all this amazing chemistry background of Everest, we developed molecules that are able to interact with a particular co-repressor of this retinoic acid receptor. So our drugs don't target the retinoic acid receptor that, as some of you might know, has too many things. I mean, it has too many functions, but just a very discrete part of the program that is controlled by a co-repressor. So when we have these novel modulators of this receptor, and we look at CMS, so this is the molecule, one of the many that we publish. So this is a first-in-class molecule. As I say, it takes out this break of the retinoic acid receptor. We have done a lot of medicinal chemistry, so they cross very good properties. They cross the BBB. They have very good biodistribution. They don't have toxicity. This was daily administration for 6 months, and now we are already in 10 months, and the animals are still quite healthy. And then when we look, we wanted to know to make sure that they were really activating CMA. So if you remember, we have these fluorescent animals. So if we gave them the compounds, and you look, for example, in the liver, you have very beautiful amount of dots. So your lysosomes are being activated. And look in the brain, these are normal neurons. And now look the amount of CMA, each of these dots corresponds to CMA activation. So, in a young, healthy animal, these drugs are able to activate CMA. So what's happened now is in a disease, age-related diseases. So Mattia in the lab, and we have done many diseases models, but just to use this one as an example, he used frontotemporal dementia because we have previously shown that CMA activity gets very compromised in neurodegenerative conditions, but mostly front tauopathies and also synucleinopathies. So we have done in both, but in this case, we use a mouse model that reproduced frontotemporal dementia. And then when he gave so we will have the wild type animal, the animal that reproduced the frontotemporal dementia, or at least the toxicity, and then with the animal that we gave the drug. So one of the characteristics of the model is that they present hyperlocomotion. As you can see here, this is just tracking their movements. So just being in the drug normalize that. The other is that they lose memory. So as you can see here, this is the Y maze. So they have less memory. But then when we have them with the drug, they preserve the memory, and they are less anxious, they are less depressed. So they have a bunch of very good traits. But we wanted to know, is this because we are clearing these neurons? And is this because we are targeting CMA? So if we look at the brain, so these are 3 brain regions. These are the healthy animals. This is the diseased model. And we are staining here for tangles of tau. So tau is the toxic protein that accumulates in the brain of patients with frontotemporal dementia. So our idea is that, okay, now we look at the brains of the animals that have received the drug, can we see a little less of these black spots, a little less of these dents? And this is what we saw. So we are very excited because, as you can see here, not only we're improving the memory and the whole thing, that's what we want at the end of the day, but we are doing it by clearing up part of this protein. So I'm very aware that this is still mouse work, but I think it's heading in the right direction. In addition of diseases of aging, we have started, and this is particularly for Neil, that he always say, what about aging, like physiological aging? So this is just all animals, 18 months, and we have been giving them this CMA activator, and then we have been looking at neuronal activity. So compared to the young animals, the old animals have hyperexcitability and seizures as you get old. And so there are microscopic seizures that you can record them. So if we maintain the animals with the drugs, we don't see that. And the same thing, like they perform very poorly motor coordination in the rotarod, but if you treat them with the drugs, they recover the ability to activate. So this is for neuronal function. As I mentioned, all animals have more senescent cells, or animals that have been treated with the drug when they were 18 months, they have less amount of senescent cells. And then, for example, if we look at the stem cells, so this is capacity of stem cells to proliferate and repopulate the bone marrow from mice. And as you can see here, treated with the drug, that is the CMA activator, they recover the ability to repopulate. And this was kind of exciting because this was in mice, but we were able to get also stem cells from old people. These are 85-year-old. And as you can see here, just by in vitro treatment with the drug, we recover the ability of reactivation of the stem cells for proliferation. So we think that even in a background of physiological aging, the reactivation of CMA is having beneficial effects. So this is kind of the summary slide. We think that proteostasis can be one of the targets for gerotherapeutics. In this particular case, I was talking about CMA, so I think it's a good target because we think it's basic to the process of biology of aging, because when it decreases and you mimic this decrease, you see many of the traits of aging and functional loss. I didn't show you, but if we decrease CMA in the context of disease, we have evidence that also aggravate diseases of aging. And then as I show you, restoring normal CMA activity, not only slow down multiple of these drivers of aging, but also it seems to ameliorate some of these age-related diseases. Regarding diseases, what are we thinking about? So we are thinking to target diseases based on what we found that happen when you don't have CMA. So it means that reactivation of CMA, because of the importance of the aging component in neurodegeneration, can be a common use for some of these neurodegenerative disorders. So far, we have worked in Alzheimer's, Parkinson, and ALS. And we have just published work also in retinal degeneration. So when you see D, that means that we have done it with the drug. So this is pharmacological intervention. And in the others, we have the genetic proof of concept with the genetic model. So we have improved sarcopenia by restoring CMA activity. We also have ameliorated some of these problems of fatty liver, atherosclerosis we published last year. We have improved bone marrow repopulation, and this was also done genetically, but also with the drugs. The Macian lab has been showing that the treatment of the pharmacological enhancement of CMA improves T cell function with age and response to vaccination, for example. And we think that it can also have a role in some of these sleep diseases of aging. So with that, I want to thank you for your time. I hope I'm still on the time that Alison was targeting, so we have time for discussion. And just to let you with the names of the amazing people that I have the luxury to work every day in the lab, and terrific collaborators in anything that is outside an lysosome. I only know about lysosomes, so anything outside that, we owe it to these amazing collaborators. And this is the people who gave us the money. Thank you so much for your attention.
Wonderful. Thank you so much. That was, really great. Thanks a ton for this awesome overview. We already have a bunch of questions here lined up. So I don't know if you want to take a brief break or if you're ready to go.
No, I'm ready to go.
Okay, wonderful. Well, then we'll make good use of time. Erin, you go.
The images that you had of the 3D images were amazing. Is that just a snapshot? Or, and then second, can you get that kind of imagery of CMA activity in the body in humans? Like, what would it take?
Yeah, that's very good. Just give me what.
I think she'll be just back, but it's also just a reminder for everyone else to put any questions or comments that you may have here in the chat so we can get to them once we have them all queued up. I know that we have Aaron, Victor, and Trion next. And perhaps we can see if Anna remembers the question.
Yes, so the
and,
question
then you
is
can
like
go for it.
those snapshots, right, of the image that I showed of our fluorescent animal. So that was a snapshot, but yeah, just to show you. But as you saw, I mean, we do 3D reconstitution and movies, and this is very important. You make a good point because in different part of the brain or depending on the organ, it's very important to know what is changing. So, for example, even in something as boring, some people might think the liver is very boring, right? It's only hepatocytes, not so much a sign in there. When you look, you have spatial temporal differences in CMA activity. And I think this is very important to see if areas more vulnerable to disease, when you have pathology in the liver, correlates with those ones in which you lose CMA earlier with aging. So we are doing as much as we can. It's kind of a mega paper because every person in the lab is expert in an organ, so they are going organ by organ, cell type by cell type. But then you make a very good point. How are we going to do it in humans? So unfortunately, in humans, we can still not put a probe like this that then you can go and get an MRI and then you will have highlighting your CMA. But something that the Mathian lab, so this is immunologists have a go on that. So what they have been doing in collaboration with us is taking cells from your blood. But the blood, of course, many cells that last very little. So the key here is to select, for example, naive T cells that they have been with you since you were born, many of them. And then basically those in animals, we have seen that recapitulate very well the changes with age in CMA. So, for example, in collaboration with Luigi Ferrucci, that you know is the director of the Baltimore longitudinal study, what they have started to look is like physiological aging, healthy individuals of different ages. What is the stage? What is the baseline for CMA? What is the baseline for other types of autophagy? And then the beauty of the study is that because it's with cells from the blood, you can do it at different times. So you can have a temporal recording of how people, their CMA or their autophagy change with age. And the other is that you can use it to see if your intervention is really having an impact on CMA. So, for example, they have a collaboration with the Michael J. Fox that they were very interested in our Parkinson studies, and they were testing one of these drugs. And the way that they were seeing if in the patients they were modulating CMA was by taking the T cells, and then the Mathian lab was looking at CMA using the same fluorescent reporters that I showed you. So as far as you can take the cells out, skin fibroblasts we have also done work with that. In the whole body, we cannot do that yet. So I'm hoping that at some point there are some people with good biophysics training that they will be able to design and biochemical design some probes that can do the same that we have right now in the animals. But we can use as surrogates cells at this moment.
Wonderful. Thank you so much. Next one up, we have all the way on top here, Victor, if you want to.
Yes, hello. Thank you for a very interesting lecture here. I just have a question. You have heard this many times before, but what do you think about the fasting and autophagy? Because I've heard that the autophagy is said to peak in humans after 48 to 72 hours of fasting, and then it would plateau. What evidence do we actually have? Because it's a bit hard to know the autophagic flux in humans.
Yeah, so I mean, we have done studies on autophagic flux in humans based on peripheral cells, and also because we were able to get the first tissue from autophagy, so then we can measure the flux. Of course, it's ex vivo, but it reflects very well what we have there. So the thing with autophagy is that you might not want it to have it up the whole time, and that's something to kind of understand. So, for example, for macroautophagy, for the other type of autophagy, the physiological way, and what we want is to return to your physiology of when you were 20, right? Because you were okay. So we want to return to that. So macroautophagy, for example, when you don't have nutrients, it peaks at 4 to 6 hours. And then basically, you eat again. So just to give you an idea, for example, if you don't snack in the middle in between lunch and dinner, your liver will activate your macroautophagy just to get nutrients, basically the great proteins that you don't need or that you can eliminate, and then get the nutrients. And then when you get food, it will go back again. So when you do these dietary interventions, even not dietary, like even rapamycin that goes through the mTOR, what has been shown is that, yeah, you activate autophagy, but then the cell finds a way to go down because that's the physiological way to go down. And you want to really heart fatigue. So the very early work that was done with calorie restriction, basically what they showed that in macro, if you calorie restrict animals, the macro autophagy ticks of up and down become sharper and nicer. It's not that you have continuously macro autophagy on, it's that you regulate, even if you are in this starvation, you just regulate the balance, the hormonal regulation of these pathways better. In the case of CMA, because it's paper type of autophagy, because I mean, just if you think of the mechanistic in macro autophagy, you are creating a vesicle. So of course you are going to trap the proteins that you want, but there are going to be also some other proteins that just happen to pass by when you close the bag of garbage. Meanwhile, in CMA, the one making the CoA is the chaperone. So you will never send to CMA something that the chaperone has not identified. This has to be eliminated by CMA. So then if you increase, and that's why we target the lysosome, rather than targeting the chaperone, we decide to target the lysosome because we just want the lysosome to be ready. Because what happened with age is that the chaperone recognizes the substrates, but doesn't have where to go because the lysosome is not ready. So we see beneficial effect because you enhance now the status of the lysosome so it can receive more, but you will never take anything that you don't need to degrade because the chaperone is kind of the police there. So in that case, it's safer. So for example, these animals that have this extra copy of LAMP2A, they will maintain CMA activity basically higher, like basal CMA activity is higher than the ones that don't have it. But then if you do a stress, they can still go higher. So there are other mechanisms that can contribute. So that's why, for example, with this calorie restriction, what you can see increase continuously will be CMA. As you point out, it has been shown that macro autophagy plateau, but it also goes down. But I don't think that's a bad thing. You probably want this up and down. Victor, I'm not so sure if I have clarified the idea, but the idea is that you don't want to have macro autophagy continuously on the whole time.
Yes, thank you. So do you recommend people to do a prolonged fasting once in a while to extend their life? Would you?
Yeah, so I shouldn't give any recommendation because I studied medicine and I never practice because I don't want to kill anybody. So just take it as friends talking in a coffee shop. So I'm not recommending anybody anything. But if I would have to think about something, I like the food clocking, this 8:16 biologically for me make a lot of sense because in the 8 hours that you don't eat, you will activate macro autophagy. In the 16 part of the 8:16 that you don't need, you will activate CMA. CMA requires a little more of time, like at least 8 to 10 hours to get at the maximum. So that means that every day you're having the best of both autophagies just by doing this cycling rather than starving for several days. But, that's my personal opinion based on the work of seeing Einstein doing this in food clocking of these 8:16 hours that he used to do.
Okay, wonderful. Thank you. Next one up, we have Creon with a whole host of questions.
Yeah, so I have a whole host of questions, but let's start with this. The process that you are focused on here, how much of the pathways that you're looking at are shared with other autophagy processes, and how generalizable is this mechanism?
Yeah, great question, because actually I will make a case. I had another slide, but I took it out. I will make a case that you need to know which autophagy you are going to target, and you might not want to target them all. I mean, in part is what I say, like CMA seems very safe. So the best thing is that you target one, and if that one improves, the others indirectly are going to improve, because it's the same thing. If you used to have only, one, like 3 people cleaning and 2 of them don't work anymore. So at least if you recover one, the other start having less thing to clean. So it will work better. But I think it's very important, and this is why I'm very happy that now, initially when the autophagy explosion, everybody wanted to activate autophagy, didn't matter what, or how, or where. And now there are many of the biotech companies are very focused, either they want to activate mitophagy, a type of macro autophagy, or they want to selectively target that pathway. We are selectively targeting CMA. And I think it's very important to know what you are going to target. What we are still missing, and the reason why we went for CMA, is that in CMA we have this global reduction of LAMP2A, that we have seen in many organs. It's a very simple process. It's very easy to target. Well, very easy. It took us 7 years, but at least we know how to do it. And there are other ways that can be done. In case of macroautophagy, the machinery is much more complicated because you have to form these autophagosomes. If you form too many and the lysosomes are not ready, they will accumulate. So there are many steps out there. There are some successful drugs that they are still experimental, but they look very promising. But it's not as simple as for other processes that is linear. So I think, selectivity on what you target, and also always look at the other autophagies to see what happens when you target one, is going to be important, because so far we have found that you block one type of autophagy and the others try to compensate. It's never perfect, but at least they try to compensate. We have never seen, and this is, I think, biology at its best, we have never seen that by increasing one type of autophagy, the others will become lazy and work less. So it only works in the good sense. It's like if you don't have one, the others will take the load. But if one starts working more, the others are still in their working. So I think that's good news for therapeutics, because that means you can enhance one, and in theory should not have a problem for the others, and should not decrease the others. But I still think that we should always look at that, because you never know if what I'm talking so general is really as universal as I'm pretending. I mean, how many cell types we have looked? We haven't looked at every single organ. So I think for therapeutics, it's always very important to keep that in mind, that always look at the rest of the proteostasis machinery for any intervention.
Yeah, that is good news. So I'm going to bundle all the rest of my questions into one last one here, one follow-up. Do you have any insight, or are you working, or is anyone working on understanding this throughout the life cycle of an organism from development through aging? So these pathways that you're elucidating and manipulating, are they active in development or not? And do they deactivate in aging? I believe you think they do. Do long-lived species keep these pathways active for longer? That's over.
So that's great questions, all of them. So unfortunately, I only have like 20 people, and we do as much as we can. But development, definitely we have done, and this is, please take it with a pinch of salt, because it's not even published yet. But what we have seen, so this gene, this LAMP2A, is a splice variant. And I think that's the coolest thing in Earth, because what happens is that with the same gene, you can do LAMP2A, LAMP2B, and LAMP2C. And it turns out that A is for CMA, but B is used for macroautophagy, and C for RNA degradation in lysosomes. So what we have seen in development, and it's really a beauty, this is collaboration with Mark Meller, is that areas that are still under development, early in development, for example, if you think on the brain, the ventricular area, you have a lot of macroautophagy. So you have a lot of LAMP2B, because that's what they need to do all this major remodeling and become real neurons. Once they differentiate into neurons, like, pluripotent, multipotent, tons of macroautophagy, very little CMA, then as they start with more specialized differentiated cells, even astrocytes, glia, and neurons, then you see kind of this gradual shift. They still will have macroautophagy, but the amount of this LAMP2B decrease, and you change the splicing to have more LAMP2A and more LAMP2C. And it's kind of like, black and white. You can really see these differences in the developing brain. It's kind of this. So something that we are very interested right now is like, who regulate that splicing? Because maybe this way we can also in aging force a little more of the LAMP2A as another way to do it. But definitely there are very important pathways in development, but they change the contribution in each cell, but all the three pathways, all the different types of autophagy remain active, at least in mammals. They all remain active past development in adult organisms. And then regarding the decrease, so I have focused today in CMA. We have also published decrease in macroautophagy. The kinetics and the timing seems to be different depending on the organ and the cell type. Unfortunately, we just started with a similar fluorescent animal, but for macroautophagy, and the animals are still aging. So I don't have the data for every organ as we have for CMA, but I think that's going to be important information for targeting and therapeutics. And then for the other extreme, long life. So there is very elegant work from Viviana Perez. She published, they couldn't measure CMA per se, because at that moment They didn't have the tools, but they inferred it by the levels of LAMP2A, that, as I say, is our key protein. And it turns out that the naked mole rat has higher amount of LAMP2A and beta proteostasis. And so, for what is worth that animals have it, we did some work with the rockfish, and we were able to also identify higher levels of the chaperones in that case. And then the Martian lab, actually in collaboration with NIR, used a NIR marginalized population of centenarians that, as you know, is a beauty, right? They have the centenarians and then the progeny with the spouses that are not coming from centenarians. And they have shown that macroautophagy, centenarians and the progeny have much better macroautophagy than the non-centenarians. And because they did it with T cells, it's really elegant, because not only does they have beta proteostasis, beta CMA, beta LAMP2A, but they can measure T cell function. So there is a linear correlation that the beta, these individuals preserve their LAMP2A or their LC3, autophagy, the better T cell function you have, the better are able to react to any immune challenge. So I think that's kind of a good, elegant proof that preservation of these autophagy pathways is, of course, in centenarians. And now that NIR has sequenced all these centenarians and has all these beautiful proteomics, he was telling me that autophagy and lysosomes and proteostasis is one of the pathways that came up on those. And hopefully we'll have a chance to data mine a little more and apply our CMS score to see how they are. Yeah.
Thank you. Beautiful answer. I'm out.
Okay.
That was a bundle of questions. Okay, great. And Lee, you're next.
Hey, great presentation. So I was wondering if you looked at proteomics after the compound treatment to see how what else is changing along with the LAMP2A and how confident you are at the mechanism of action here.
Yeah, I'm so glad you asked for that, because I mean, proteomics has been a major revolution for us, because protein degradation is all about proteins. So sometimes transcriptomics for us were like, but proteins, we can really get a lot out of that. So we have actually done that. And I love that you asked that, because when we compare our genetic model that we are only modulating LAMP2A with the animals that we gave the drug was worse better. So then we were like, well, it might just be the biodistributions and whatever. But it turns out that when you look at the drug, it's acting in more than one component of the CMA network. So as I mentioned, it's a very discrete program. So there are 32 proteins. And out of those 32 that are modulated by the drug, 13 of them are CMA components. So we really think that the other 10, they might also be components of this pathway, and we have never really studied them before. So now I have 2 graduate students trying to check each of those ones and see if they just happen to also be components of this pathway that we didn't know. But I think that's the beauty of that very selective target that Everest was able to do, and that's why the animals are better than just increasing LAMP2A, because, LAMP2A is limiting, right? But, at some point, if you have all the LAMP2A that you need, something else is going to probably be limiting. And I think with the drugs, you are maintaining really the stoichiometry of things, so you can have much more for the same price, I guess.
Very interesting. Thank you.
Yeah.
Awesome. Jason, we have you next. And for me, that is the final question from participants. So unless you want me to ask questions, feel free to put some more in the chat or raise your hand. Otherwise, it will be me with our foresight questions up.
Awesome. Thanks, Allison, and thanks, Dr. Anna Maria. Really, really great presentation and targets. I was curious about the relationship between the mitochondria and CMA and just mitochondria and autophagy in general. Kind of what have you seen when you're activating that pathway? And I'm also serious. I'm also always thinking about the relation with mTOR as well. I don't know if you have thoughts on those two, the interplay between those.
Yeah. So great question. And I love that you are in the gym, because exercise activates autophagy. So keep going. Yeah. So the relation with the mitochondria, terrific question, because one of the first things that we saw when we look at our animals that have better, we do electron to do that, and the mitochondria were gorgeous, like compared to, they didn't look like mitochondria from an old animal at all. So of course, we were like, okay, that we cannot blame to CMA, because CMA will not be able to take a whole mitochondria. The way that you renew mitochondria in your cell, as you know, the mitochondria is this big network. They get fragmented, and then they are taken by macroautophagy, by the other type of autophagy that I didn't talk in a process called mitophagy. But, our CMA cannot do that, because this is like proteins have to cross a membrane. So we found two things. So we found that even it cannot take the mitochondria as a whole, in normal conditions, as you know, many of the proteins in your mitochondria are synthesized in the nucleus. So that means they have to pass through the cytosol to reach the mitochondria. So it turns out that CMA can control, when they pass through the cytosol, can control the enzymatic load of your mitochondria, how many enzymes go there, how active your mitochondria are going to be. So I think by restoring CMA, we were improving the quality of your mitochondria. But then the other thing, as I mentioned, when we restore CMA, we also see that macroautophagy becomes better, because now it's in a cleaner environment. It doesn't have to take the whole load because CMA is doing its part. So we think that mitophagy, we have some evidence that mitophagy is also functioning better. So I think the connection with mitochondria is very clear on those two. And then regarding mTOR, as you know, this pathway is really activated. Part of the regulation of autophagy is always nutrients, right? That's how originally it was described in the 60s by the Duke. You activate autophagy during starvation. So mTOR is a major negative regulator of autophagy. If you eat, mTOR will go activated. You will not have so much autophagy. When you are starving, mTOR will not be active, and then you release the machinery to do autophagy. So that's for mTOR complex 1, TOR1. And then what we found is that, so TOR1 will be inhibitory of macroautophagy, so your rapamycin will activate macroautophagy. And then TOR2, the other complex, is inhibitory on CMA. So by acting on TOR2, you might enhance CMA activity. So we think that the balance, and it's a bit more complicated because also CMA regulates mTOR, so it kind of arrows in all that directions, but we kind of like these vicious circles, right? Like if you have something that self-regulate and regulate the other, if you act in one, you restore CMA, hopefully you will also restore mTOR signaling, or if you restore macroautophagy, because you maintain homeostasis there. But I think it's a complex relation, but in principle, mTOR is inhibitory of these pathways.
Just a quick follow-up. I mean, if you're taking something like metformin, which is, looking into mTOR, I mean, would that, if it's decreasing mTOR, would that increase your autophagy potentially?
Yeah, so we have a paper, and the credit goes clearly to Derek Huffman, that he was looking at different components like sarcosine, and we include also metformin, and metformin activates macroautophagy. So I think it's probably, we didn't follow the mechanism in that case. I think there are other studies now showing that, but it's most of these therapeutics that improve lifespan, and they act in different pathways, and many of them are activators of autophagy, like spermidine. You probably have heard about the spermidine, acarbose, metformin. So they all have some degree activators of autophagy.
Nice, thank you.
Wonderful.
Appreciate it.
Thank you so much. Micah, we have you probably as the last one before I ask the final closing question.
Feel free to answer these quickly since we're almost out of time. The first one, did you do a lifespan study with the drug or only with the engineered mouse?
So we did it with the genetic mouse, and the one with the drug is ongoing. So those functional studies that you saw, those animals are still alive. So they have reached 23 months. I think that's the oldest that we have right now. And what we did is administer the drug since they were 18 months. And then we have a second group that just started at 22 months. So this way we can know if there is a point of no return. But we don't we are waiting for them to die.
Okay. And the last one, for the engineered mouse, do you believe that you successfully kept CMA active for the entire life of the mouse, or do you think you eventually lost the battle and it declined with age just later?
Question, and actually, because we had the dendro animal, these animals that are fluorescent, what we did is we crossed our LAMP2A animal with the one fluorescent, so we can track the CMA until very late in life. And, it's definitely higher than in the control, so until late in life. So we have to still, it's a lot of work to study image-wise this pathway, but at least we track it in the liver and in the hippocampus. and they are clearly maintaining preserved CMA compared to the others. And now we are doing the same. The drug animals, some of them are also with a reporter, because we want to make sure, if it's not going to work once you pass an age, we want to be the first ones to know, right? So just to know if we can do other kind of combined interventions. And that's the other thing that I think as a community, we are thinking more and more. I mean, there are many different ways to modulate proteostasis or to modulate mitochondria and epigenetics. So at some point, I think we are going to start including this as combinatorial interventions just to get the most out of these changes. So if we see that there is a point of no return, maybe that's if you combine with another driver of age improvement, you can still restore CMA. Yeah.
Okay, wonderful. Thank you so much. In the final minute, I would love to ask you just if people in this group are really excited about your work in particular, then what could they do to support it? That could be, I don't know, if you may be hiring, you may be fundraising for a project, you may be looking for collaborators, anything that I think, could go into this final action, like part of the meeting would be wonderful.
Yeah, so I think, it's like everything at the end is money, right? But I think something that interests us, there are two aspects. So one is the drug development, because I think, we use one way to target CMA. And of course, now the school has licensed it to a company, so they are following on that. But I think there are probably more ways to activate this pathway. So I think continue a little with the drug screening, drug discovery. I think the more we learn about the mechanism of why LAMP2A decreases with age. So now we know that it's because it's very unstable in the lysosomal membrane because of changes in the lipid composition. So theoretically, if you can use like lipid scavengers or other ways that you restore the normal lipid homeostasis in the lysosomal membrane, then your LAMP2A will last longer and you don't have to add it. So I think there is a lot of venues that will be very interesting to explore on that direction. And then the other thing that I'm really passionate about now is really human studies, but a little on the discussion that we were having. That the first question was about how are you going to measure this in humans? So I think we still don't even know what is a normal level of autophagy. Maybe I'm 4'11", so maybe my level of autophagy doesn't need to be as big as other people that is 3 times my size. We don't even know what is the baseline. And I think now that we have this algorithm, we have, developed, like to get a prediction, it would be very good to develop functional studies that we can really measure in humans. So then when you hit 50, you can go to your doctor and they can tell you your level of CMA and say, okay, you better start behaving and taking all these things to improve CMA. So I think human studies, because, mouse animals, we are going to continue doing a lot of animal studies, but that's something that, we can handle. I think human studies and drug development is probably where we need more help, because that's not my expertise at all.
Wonderful. Thank you. This was a really, really, really great presentation. Thank you so much for joining for being so flexible and so accommodating for our questions. We wish you all the best. And I hope it wasn't the last time that we saw you. And all of you guys, I'll see you on the 28th for an exciting launch of the biomarker consortium for aging. I think you should be receiving invites for that very soon. Thank you so much, everyone. Have a wonderful rest of the day. Bye.
Thanks a million. Bye-bye.